Happy to introduce Barbara Edwards, one of our geography majors. She's a junior, borderline senior, I guess, aren't you? And she's done some very interesting work collaboratively between both the geography and the biology department. So, Barbara, it's all yours.
Okay, thank you. Well, I'm Barbara Edwards. As he said, I'm a junior geography student. And just thank you for introducing me, and thank Dr. Triplett and Dr. Scott and Dr. Gregg for helping me and me for helping them with this project. So our project is the quaternary contraction and expansion of biomes and the creation of distinctive populations of Arundiaria gigantea. So Arundiaria gigantea is also known as river cane, and it's one of the 3 species of bamboo family native to North America, the other 2 being Arundiaria appalachiana and Arundiaria tecta. It's distributed widely throughout the southeastern U.S., and it used to be in large cane breaks, but nowadays you only find them in isolated stands. We have some pictures. The one on the left is Dr. Gregg holding one of the specimens we took along the Tennessee River. And so our hypothesis is about how has the contraction and expansion of biomes produced by climate change during the late Pleistocene and present created genetically distinctive populations of Arundiaria gigantea. And so during the Pleistocene, during periods of ice advance, biomes were pushed to the south. As you can see, very little ice up at the top, and then it expanded and squished everything down. And so as it would have been pushed down south into a relatively narrow band along the Gulf Coast, you can see that right now where it would be is in the orange, and the tan is where it would be 10,000 years ago. Now, as it warmed back up, it would have extended its range back up to the north, but things obviously would have gotten in its way. So what would have affected it? And the thing about these bamboos is that they travel through their rhizomes, not with the spores or seeds. So they're not going to be carried away by the wind to faraway lands. They have to travel through the soil. And so it needs soils at least 40 centimeters thick, which isn't a grand amount of soil, but it's not going to be able to travel over mountains with their shallow bedrock. And it also needs relatively wet areas with moisture, so dry uplands are also going to be a barrier. So in summary, you need not mountains because those are going to be barriers, and they'll be paralleling major streams. And so we were looking around the Coosa and the Tennessee watersheds, which the Tennessee is this top yellow one here, and the Coosa light turquoise. And so where they do touch, separated by the Appalachian Mountains, is going to be very unfavorable because it hits all those barriers with the dry uplands and mountains. And so this is where it leads into the island biogeography, because when you have populations of plants that are separated for large, or anything really, when they're separated for large periods of time, they tend to become genetically distinct. And because these giganteas would have been separated for over 10,000 years, we hypothesize that they're going to be genetically different. And so how do we know? How do we find out that they would be genetically different is you would use morphology and kind of how it looks and everything. But nowadays we would use DNA analysis. And we would use this genetic fingerprinting using AFLP, the amplified fragment length polymorphism. And how we do this is we use the restriction enzymes to chop up the DNA so we have fragments of it. And then we use polymerase chain reaction to create enormous numbers of the particular fragments. And so what happens is you have your DNA, and then you use heat and you heat it up so it'll unzip itself. And then you have your target genes and you attach primers so that you can connect them, and then you'll have 2 copies of the gene from the one strand, and then you just keep doing this over and over. So you'll have millions of copies that you can work with. And then once you have all these copies, you can use electrophoresis to create a banded pattern that can be used to compare the DNA of different individuals. In electrophoresis, you have the fragments pushed by an electric field to create that banded pattern. And the smaller ones are going to move faster and further than the big ones. And with it, you'll end up with something like this, where each column is representing the DNA of an individual, and where it's very bright green, that's where you're going to have a lot of a particular fragment, and the dark is where there will not be very much. And so once you have this electrophoresis all analyzed, you can create a phylogenetic tree where you'll be able to show the evolutionary relationships between the bamboos, and an early divergence in the branching will suggest distinctive populations. And this is an example of the phylogenetic tree for all three species. So you can see how they're separated and grouped together as Gigantea, Intacta, and Appalachians. And so our methodology is what we did is we went out and we collected DNA samples, as well as some we have here in the herbarium within the Coosa and Coosa watersheds. And that's just some field work that we did. And so we prepared the samples for the DNA analysis, and then we statistically analyzed them to see if there were in fact significant differences between the watersheds. And our results are actually so far inconclusive. We don't actually know quite yet. So more research needs to be done to either prove or disprove it. And that is the end. So if there are any questions, I'd be happy to answer.